This is our cellular respiration stop-motion video. Most of cellular respiration takes place in the cytoplasm or the mitochondria. The first part of the mitochondria that we labeled is on the top, and it's the outer membrane, which contains transport proteins that help to shuttle pyruvate from the systole.
Next is the inner mitochondrial membrane, and this is where electrons move down the mitochondrial membrane by NAD and FAD, and it is also the location of oxidative phosphorylation and the electron transport chain. This is also near the inner membrane space, which is a small space between membranes that maximizes the hydrogen gradient.
The cristae are labeled on the bottom, and those are folds in the mitochondria that increase the surface area to volume ratio. The final part we labeled is the matrix, which has a suitable pH and the correct enzymes for the Krebs cycle to take place. The first step of cellular respiration is glycolysis, which occurs in the cytoplasm, and is a process in which glucose is broken down in 9 steps. It starts with a 6-carbon sugar. The first step of glycolysis is 2 ATP being invested. The 2 ATP are then broken down into ADP and a phosphate group. The phosphate groups are added to the glucose molecule, which means that it is phosphorylated and it is easier to break apart. The next step is the 6C sugar is broken into 2 3C sugars. In the process of oxidation, hydrogen atoms from each of the 3C sugars reduce NAD to NADH plus H+. At the same time, ATP is formed in substrate level phosphorylation. Glycolysis results in 2 pyruvate being formed, as well as 4 ATP total. If oxygen is available, the pyruvate is then transported to the mitochondria to undergo aerobic respiration.
The link reaction, also called oxidative decarboxylation, occurs in the matrix of the mitochondria, in between glycolysis and the Krebs cycle. Carbon and oxygen are removed from the pyruvate in a process called decarboxylation, which forms a carbon dioxide molecule as a byproduct. The 2C compound then forms an acetyl group when 2 hydrogen atoms are removed via oxidation. The hydrogen carrier NAD+ becomes NADH plus H+. The acetyl group then binds with coenzyme A to form acetyl coenzyme A, which then moves into the Krebs cycle.
The Krebs cycle is a process that completes the breakdown of glucose, and it occurs in the mitochondria as well. It starts with the acetyl coenzyme A molecule. Acetyl coenzyme A transfers its acetyl group to a different 4-carbon molecule to make a 6-carbon compound called citrate. A series of many different reactions break down the citrate. First, multiple oxidation reactions occur. NAD becomes NADH and FAD becomes FADH2. Through the process of decarboxylation, 2 carbon atoms are released, and they eventually form 2 molecules of carbon dioxide. 1 molecule of ATP is produced in this process through substrate level phosphorylation. The intermediate oxaloacetate is formed, and it binds once again to another acetyl coenzyme A, which forms the citrate. The cycle is then able to restart.
Next is the electron transport chain, which starts in the matrix of the mitochondria. First, the FADH2 and NADH that were produced in the Krebs cycle are still in the matrix, and these products go through the electron transport chain itself, which is made of proteins that act as a series of electron carriers, and it's located in the inner membrane of the mitochondria. As these molecules move through the ETC, they go through a series of redox reactions because of the electrons that are also moving through the ETC. As these electrons pass through the ETC, they lose energy, and this energy is used to pump hydrogen ions through active transport from the matrix to the inner membrane space, because there needs to be a maintained proton gradient between these 2 spaces of the mitochondria. And finally, at the end of the ETC, O2 will accept any excess hydrogen ions or electrons to form water. And the last step of cellular respiration is chemiosmosis. So the hydrogen ions in the inner membrane space flow down the concentration gradient using passive transport through a large integral protein. And this protein contains ATP synthase, and this is where the oxidative phosphorylation stage comes in. So as the protons diffuse through the protein, they're also diffusing through the ATP synthase, and they serve as the energy to allow the ATP synthase to join ADP and phosphate, creating ATP. These final stages of cellular respiration are when the bulk of the energy is produced, because 16 ATP are produced per pyruvate, meaning 32 ATP are produced per glucose. And that concludes our explanation of cellular respiration. Thanks for watching.